12 today. So that'll be cool because we're going to start getting into organisms that can cause disease. So it should be a lot of fun. So chapter 10 is on classification of microorganisms. We're going to talk about how scientists study microorganisms and put them into groups. This hopefully will be helpful for those of you taking the lab now, or if you're planning to take the lab in the future. The information that we cover in here about how we classify microorganisms is basically the same thing that you do in the lab in order to classify your unknown microorganisms. So when we talk about studying the relationships between species, there is a whole field of biology that's called taxonomy. And the goal in taxonomy is to be able to classify organisms into groups. People like things that are organized, and so it allows for easier study when we have a common system that we use in order to classify microorganisms or any organisms. Let's not, not just microorganisms, all organisms. Those groups are called taxa. Okay. And again, if you're a person who's interested in biology and you're also very organized, this is a really popular field, taxonomy and systematics, like studying these types of relationships and putting organisms into different groups. So we do this based on degree of similarity. And for the longest time, it was, well, this thing flies and has feathers. That thing flies and has feathers. Those are birds. They are together. Right? That's about as good as we got. Now we are able to use genetics to help really tease apart these interactions. The scientific rationale behind this is that organisms that evolved from a common ancestor likely have common features. Again, this is where sometimes without having genetics, you could put organisms together because they look very similar, but they're actually very, very different. But what we know is that all organisms, and when we talk about viruses, I think starting next week, we'll talk about why viruses don't get to play in the tree of life. But when we talk about all organisms, the prevailing scientific theory is that we all evolved from a single common ancestor. That common ancestor was composed of one or more cells, one cell, right? A cell that had a plasma membrane. So that's why, theoretically we talk about organisms that don't have a cell, they're not alive. They use ATP for energy. This is one area where students will sometimes get a little bit mixed up, even in advanced microbiology classes. They'll talk about something like a virus not being living because they'll say, well, it's dependent on its host, right? A virus absolutely requires a host. So do lots of things that we consider living. Lots of bacteria, lots of multicellular parasites absolutely require a host, but we still consider them living because what they need from the host is ATP. They still use ATP for energy. Viruses don't use ATP. They use host machinery in order to replicate. The host machinery uses host ATP. And the genetic information is DNA. A lot of viruses actually could meet this criteria. Their genetic material is DNA. But there's a whole mess of viruses that also use only RNA instead of DNA. So viruses don't get to play on the tree of life. Okay, so when we're studying these phylogenetic relationships, right, that systematics I talked about, is that study of the evolutionary relationships. So again, for people who are interested in evolution and interested in classification of organisms, there's a lot of research here. We don't know everything. We still learn new things all the time. And believe it or not, we still find a lot of new species. And you might be like, well, yeah, new bacteria, big deal. But we find new animals as well and new plants. So whatever you're interested in, we do find new ones all the time. Sometimes we think we have one organism and it turns out they're actually two separate species. And so you find out that there are new species. So these types of fields still have a lot of change. And that's why I skipped chapter 11. Chapter 11 is all about the taxonomy of bacteria. And even though they have a new edition of the textbook, it's still out of date. So, because it changes all the time. Organisms are a product of their genetic history, right? The changes in the DNA, the mutations that have accumulated over time that have led to changes in the protein and therefore changes in the phenotype of the organism give us the organisms we have. Theoretically, every organism that reproduces gives rise to an organism that is very much like itself. With the exception of like cloning, there are going to be genetic differences between parents and their offspring. In organisms like us that reproduce very slowly, after tens or hundreds of thousands of years, you may start to see things like new traits that could lead to new species. But even as recently as 40 or 50,000 years ago, there were other species of humans on the planet. Okay, so it takes a long time. We wouldn't see it in our lifetime, but it does happen. And that's why each species does retain some characteristics of the ancestors. Some single-celled organism is our ancestor. And you might think, well, we don't look all that similar, but we have a lot in common. We still retained a lot of information. The more recently we split, the more things we might have in common. But after billions of years of evolution, we may not look very similar at all. So this idea led to the theory that organisms all evolved from a common ancestor. So when we combine this idea of taxonomy and phylogeny, it can be really helpful for us to do things like identify new organisms or identify an unknown organism. We can put it in the context of what we know about various relationships. Again, with multicellular eukaryotic systems, this is a lot easier. It has 3 body parts, has 6 legs, a pair of wings, and a stinger. It's probably one of the hymenopterans, right? You can start to take your unknown organism and put it into a group. It's a little bit more complicated when we talk about bacteria because they're unicellular. There are lots of gram-positive cocci, right? But at least now I know it's a gram-positive coccus, right? I can start to put it into the group where it belongs. Okay, and so for classification, we can put organisms together based on their degree of relatedness. And the idea here is that we have what's called nomenclature. It's the common naming system that allows scientists all over the world to communicate. Every species has its name, and we talked about this earlier in the semester. And, in every language, there's going to be a word that refers to a species, right? Like a dog. And in a lot of the Romance languages, maybe the word is similar. But in other languages, it's not similar at all. So how do we talk about a dog? Well, we talk about Canis familiaris, the dog. So it provides that universal name that allows us to communicate. And so what we want to do is group organisms according to their common properties. And of course, the old method was the fossil record. And if you're at all interested in fossils, you should talk to Dr. Perlow. Her research is really cool. Not dinosaurs. My kids were disappointed, but fossil mammals. So we could use the fossil record. We could also classify things based on their morphology and their physiology. And this worked for bacteria too. It's a gram-positive coccus and it's catalase positive. We put all those together. So that's part of its physiology. More recently, I guess maybe not even all that recent anymore, but we also incorporate molecular biology, looking at the DNA, the RNA, the proteins. And this can help tease apart some of the relationships. It can help clarify relationships, and it could highlight relationships that we didn't realize actually existed. And one of the other cool things that we can do when we look at the DNA especially is we can start to estimate how long ago two species might have diverged. And we use what's called the molecular clock. We look at changes in the DNA, and this does operate under one big assumption. The assumption is that all changes in DNA occur at a relatively consistent pace, which we know is not necessarily true. There might be maybe exposure to some chemical that causes a more rapid rate of mutation. And that's why you get a range. You can't say, oh, it was exactly 153 million years ago that these two species diverged. Could be 100 to 170. Who knows, right? You come up with a range. But it does allow us to make predictions as to when species might have diverged. Now, the challenging thing, if you do want to do molecular biology, is that when you're trying to take a group of organisms and classify them and their relatedness to one another, you have to pick a gene or genes present in every organism of interest. So you can't pick a human-specific protein if you're wanting to look at mammalian evolution, because other mammals don't have a human-specific protein. If you're wanting to look at all bacteria, you can't look at catalase because not all bacteria make catalase. So you have to pick a gene that's present in all of the organisms you're interested in studying. But you also, especially if you're looking at bacteria, want to look at a gene that is not likely to be transferred, via horizontal gene transfer, because that can complicate your analysis. The gene might look identical in two very different species if two very different species have shared the gene. It gets very specific what you have to do here. It should be well conserved, meaning that the gene looks like the gene in all of the different species, but it also should have changes. Because if the gene has no changes, if the DNA sequence is the same in every species, how do you tell the evolutionary relationship? There's no change. And it has to be big enough, large enough to provide information. So we're usually talking over 1,000 base pairs is good resolution. And I look at all those criteria and I'm like, ugh, how do you pick a gene? Especially if you want to study everything. I want to study every living thing. How do I pick a gene? Well, rRNA. rRNA works because rRNA is well conserved. Every organism has rRNA. They have ribosomes. They have to have rRNA. Bacteria have 3 different kinds of RNA. Or rRNA, excuse me. They have 5S, 16S, and 23S. 16S is most common that we use. 5S is too small. 23S, it's bigger. It just costs more money to sequence something that's bigger. So we tend to settle for the 16S rRNA. Eukaryotes have an 18S rRNA that you could use for eukaryotes. But again, commonly 16S, but 23S is also done sometimes. The other thing to remember is that eukaryotes, while they don't depend on 18S rRNA for, building their own ribosomes, there is a little part of them that has 16S, their mitochondria. Okay. So you can use it. And the 18S and 16S can be, again, used to do this type of classification. It is well conserved, but it does have enough changes that you can actually see the difference between two different species in the same genus. Sometimes even two different strains of the same species. So this idea of using rRNA in order to classify all living organisms is what gave us our 3-domain system. So it was developed in the late 1970s. So like I said, it's, not really modern anymore, but it's more modern than biology history. And this actually didn't make its way into textbooks for quite some time. Even when I was going to like junior high and high school, we didn't talk about a domain system. We talked about 5 kingdoms. And so they put all prokaryotes together. So anything that was a prokaryote was together. All of the unicellular eukaryotes were together. Then there were fungi, plants, and animals. That's what they taught us. And I'm like, cool. And then I get to college and they're like, actually, there's 3 domains. I was like, well, cool. That's why I came to college. I wanted to learn new things. So I'm old, but I'm not that old. So this is relatively new. And it finally made its way into textbooks, and this is the well-accepted idea in biology. Our 3 domains, right, are the bacteria, the archaea, both of which are prokaryotes, and then, of course, our eukaryotes are their own domain. And, we illustrate it in this tree of life, but of course there are a couple of complications in the tree. We have the bacteria that gave rise to the mitochondria that jumped over here, the bacteria that gave rise to the chloroplast that jumped over here. Who knows what other kind of exchanges were going on. And so kingdom was not the top classification anymore. Domain became the top classification. So now you have like domain, kingdom, phylum, class, order, family, genus, species. And then they get really weird. See, I'm not a taxonomist. You can tell I say it's weird. And infra-class and supra-order. I'm like, oh my God, y'all. I leave that to the taxonomists. I only know the big ones. So when we look at these 3 domains, oftentimes beginning scientists, so not you guys, beginning scientists would assume that bacteria and archaea are probably most closely related because they both lack a nucleus. It's a big fundamental difference. So clearly they are more closely related to each other than either of them would be to the eukaryotes. And what we find is that it's a lot more complicated than that. It's not a simple, who's closer to who. So yes, they do differ in cell type. Bacteria and archaea are both prokaryotes. The eukarya are eukaryotes. They each have a different kind of cell wall. If they make a cell wall, bacteria have peptidoglycan. Archaea usually have pseudomurin, but no peptidoglycan. Some eukaryotes don't even make a cell wall. When they do, it's usually a carbohydrate derivative. But then we start to get kind of more into the weeds about these organisms. Archaea have a membrane with an ether linkage. And I'm not going to expect you to remember this. But bacteria and eukaryotes have one with an ester linkage. So based on membrane lipids, actually bacteria and eukaryotes are more closely related to each other because the archaea have a very weird one. Then when we look at the first amino acid and protein synthesis, archaea and eukaryotes use methionine, Bacteria use a modified methionine, formal methionine. So in terms of protein synthesis, archaea and eukaryotes are more closely related to each other, and the bacteria are different. Sensitive to antibiotics, only the bacteria, so archaea and eukaryotes are closer. A loop in the rRNA, only the bacteria. A common arm in the tRNA, the archaea lack it, but bacteria and eukaryotes have it. Again, I'm not going to ask you to remember these differences, but what I want you to take home from this is that this idea that bacteria and archaea are kind of this conglomerate of prokaryotes and they are so different from the eukaryotes is not always true. At that molecular fundamental level, there are situations where archaea are closer to eukaryotes, or bacteria could be closer to eukaryotes. So the evolutionary relationship is just a little bit more complicated than we thought. And then, of course, we have our classic taxonomic hierarchy. You may have learned one of any number of mnemonic devices to remember the order. Okay, but domain, kingdom, phylum, class, order, family, genus, species. That's just the order. It kind of is what it is. Most of the time in prokaryotes, we do not use the kingdom designation. We skip straight from domain to phylum, but that's okay. So it gives you some of these different organisms and then all of their different levels of taxonomic organization. Okay. And again, I'm not going to expect you to remember like, oh, baker's yeast fits. Like, come on. Just again to show you that these levels exist. So with this taxonomic hierarchy, as I'm sure you know, the species is the most basic unit. And a lot of the way we define a species is very eukaryotic centric. Organisms that are capable of interbreeding are the same species. If they cannot interbreed, they are different species. But then we have to qualify that, because if you put a horse and a donkey together, they can have a baby. But we think horses and donkeys are different species. So then it's, if you put them together, do they have a viable offspring that is also fertile? Mules are technically infertile. Right? You can put a tiger and a lion together and get a liger. Why you would do that, I don't know, but you could. And then you can start talking about other organisms where it turns out that we call them different species, but they're capable of interbreeding and their offspring are capable of breeding. So eukaryotes are weird. But in theory, that's what we say. If they can interbreed, technically they are the same species. If they cannot, they are different species. And prokaryotes, what do? Because prokaryotes don't breed. You put two bacteria together, they're just going to replicate on their own. They don't need each other, they don't care. So prokaryotic species are more difficult to define, and it's a little bit more arbitrary. It's a population of cells that have similar characteristics. And now we, of course, add genetics in there as well, and there's arbitrary genetic cutoffs. We talk about cultures, which are bacteria that are grown in a laboratory setting. We talk about clones derived from a single parent cell. And we can talk about strains that are genetically different cells within a clone. How does a clone become genetically different? Remember, DNA polymerase isn't perfect. Every time a cell replicates, it can accumulate mutations. You leave a culture long enough, the cells will technically be genetically different. Bacteria are hard to work with that in that way. Okay. So again, what I'm looking for here is that you could define that a eukaryotic species, most of the time we're talking about they are the same species if they interbreed and produce viable offspring. They are different species if they cannot. With prokaryotic species, we're looking for similar characteristics. And in just a little bit, we'll talk about what some of those characteristics are. And then just to bring up these guys that don't actually get to play in the tree of life, we have our viruses. They are not classified into the 3 domains. They are not composed of cells, and they use the machinery of the host to multiply. Even the most desperate, obligate intracellular bacterium has its own ribosomes. It has its own DNA polymerase. It may need the host energy to run that, but it has its own. Once inside the host cell, it does not take the host cell's ribosomes or polymerases to replicate. It might steal the ATP and other nutrients. But sometimes people will talk about a viral species. And so a viral species is a population or a group of viruses that have similar characteristics, and they occupy a particular ecological niche. Perhaps they bind to the nervous tissue of mammals, like rabies. Or they can bind to the respiratory tract of multiple species, like COVID or the flu. But we'll talk a lot more about viruses. Okay. And again, we've talked about this idea about scientific nomenclature. So just to really emphasize how important this is, I teach a graduate class, and one of the graduate students did the nomenclature wrong, and I'm like, oh God, graduate students, you gotta do the nomenclature correctly, okay? You have to use both terms. We talked about this already, because there are lots of things that end in coli. A lot of things that live in the colon end in coli. There are also multiple members of the genus Escherichia. So you just use both names. Okay, questions to this point about classification, taxonomy, before we really get into how bacteria are classified into different species? Okay. All right, so what do we do with bacteria? Well, it's a little bit more challenging because we can't just look at wing length or scale pattern, right? We have a single cell. So we try as much as possible to compile all of the information we can and then group the organisms together in that way. Clearly, a gram-positive coccus, a gram-negative rod are not close relatives. So if we find a gram-negative rod, we look at our other gram-negative rods. And then we look at, is it oxidase positive or, whatever other assays we would use. And so what you do in the lab, right, the big project for the second half of the semester is you're given a mixture of 5 bacteria, and you have to match those bacteria to the list of possible bacteria. So you have to identify an unknown organism based on its properties. This is also really foundational for any of you interested in careers in clinical lab science. So if you're wanting to pursue CLS, it's really important that you're able to take a patient's unknown sample and identify what infection they might have. So what can we look for? I've used, of course, the idea of shape, gram-positive, coccus versus bacillus versus spirochete. Those are all probably pretty different. We could talk about size. Like if you look at B. megaterum under the microscope next to E. coli, yes, they're both rods, but B. meg is huge. E. coli is like this little tiny rod. So the relative size. Does it produce an endospore? There are lots of gram-positive rods. Not all of them produce endospores, but bacillus does. Does it have flagella? Things like E. coli and Proteus are really motile. They usually have flagella. Things like Pseudomonas might only have 1 or 2. Does it have a lot? Does it have a few? Does it have any at all? Klebsiella usually doesn't have any at all. So flagella. And then, of course, differential staining. Well, it's a rod. Is it gram-positive or gram-negative? Or the same thing with the coccus, gram-positive, gram-negative. It's neither. Is it acid fast? So that's based on the different chemical composition of the cell wall. And then, are you into the biochemical tests yet? In the lab? What are you guys doing in lab this week?
Results.
From
the biochemical.
Oh, from some of the biochemical. Okay, cool. So you are working on the biochemical assays. Very cool. So the biochemical assays, right? You can't just do one, you do a whole suite of them, and then you can really start to find out, okay, I have a gram-negative rod, it's motile, it ferments lactose, it's oxidase negative, blah, 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 it's E. coli. You can start to make those calls about what unknown organism you have. Now, hopefully those of you who go into clinical lab science won't have to actually do like all of the individual, I'm going to do this carbohydrate, that carb, like all the different tubes and tests. Hopefully your lab has money. If your lab has money, you get these. These are called interrotubes. These are really cool. If you take medical microbiology, you do get to do this. You don't have to do all the individual biochemical tests. There's 15 different biochemical tests in this one tube, which is much nicer than having to do the 15 individual biochemical tests. And so you can test for glucose fermentation, gas production, lysine and ornithine utilization, hydrogen sulfide, indole, another carbohydrate here, lactose, arabinose, sorbitol, VP, phenylalanine, urease, citrate. Nice, right? You're like, well, I just did like 10 of those in the lab right now. So you do it all in one. You have a little needle. You pick your colony and then you just kind of pull the needle through to inoculate each little chamber. You incubate it. There's a couple of them that have a little plastic, almost like a little saran wrap that you puncture and add, like for VP, you add the little reagent so you can get the VP results. And then they group them into like groups of 3. And then so left, middle, right are all 4, 2, 1, 4, 2, 1, 4, 2, 1. So the first group of 3, if it was positive here, right, 4 plus 2 is 6. Here, only this one was positive, so 2. These 2 were positive, 3, right? You get the 5-digit code, and then you have it used to be a book, but now you have a website, and you enter that in there, and you know what species you've got. It's really cool. I suffered through the same bacteriology class you did. I took this class. Those of you who have Dr. Tao, I had Dr. Tao. I've done it. I've been there. We only had to do like 2 species in our unknown mix. She got harder over time. She says it was 3, but I'm pretty sure it was 2. But now it's 5. But when I taught micro at University of the Pacific, they only did this, and I was like, this is magic. She had never told us about it. I'm telling you about it so you can take med micro and then get to play with the magic. So that's a really great way to do 15 different biochemical tests and much more easily identify your organism. One of the other things that we can do to try to identify organisms is look at the patient, especially looking at either their production of antibodies against an organism or looking at known antibodies and seeing if the patient has antigens against those that work against those antibodies. We call it serology. Serology is really useful for diagnosing viral infections. Like an interrotube is not going to work on a viral infection because you can't culture the viruses. So what you look for then is either the fact that a patient is producing antibodies against a potential infection, or you test whether or not the patient has an infection that reacts with a known panel of antibodies. I think we've all had experience with this. Who has taken a COVID test in the past 6 years? Yeah, okay. Right. Now the tests are real fancy. You can go to CVS, because I had to do this a couple weeks ago. You can go to CVS and get one that tests for RSV, flu A, flu B, and COVID all in one, which is great because I only had to swab the kid's nose like once because she got really irritated when I started doing that. How does that work? Like, how does that test work? The way it works is that there are antibodies against those different viruses inside of the tube, or inside of that little, flat thing, whatever it is. You, right, get in there and you see if you would pick up viral particles, right, when you do that. You mix it with the solution, you drip it on there, it moves across, and when the antibodies in the little test react with potential antigens being produced in the nose, you get a colored band. It's a colorimetric assay. So you're using serology at home to diagnose yourself. Not me looking at my kid going, flu A? Are you kidding me? We were vaccinated. Everybody got it except me. Unlucky for you, because I didn't feel sick. I got to come to work. So the idea here, right, is that you can look for antibodies. This is also really common for like first-line testing for things like HIV. You take a patient's blood sample and you look for the production of antibodies against HIV, because you don't make antibodies against HIV unless you have HIV. So these types of tests using antibodies are really, really useful for identifying what type of infection a patient might have. I knew something was wrong with my kid. I didn't know which respiratory infection it was. It was flu. Now I know. Okay, so that's how those tests work. When you're specifically trying to look at bacteria, this isn't terribly common, more in a research setting than in a clinical laboratory setting. You can use phage typing. So you take a known phage, or you take a phage cocktail, and you see whether or not the phage are able to infect the bacterium. If they are, you can figure out what bacterium it is. Phage are usually pretty specific. It infects E. coli, it infects E. coli. No infection, no E. coli. So again, not terribly common in a clinical setting. Sometimes you can use it for disease tracing or outback tracing, but outbreak outbreak tracing. Jeez. Okay, there it is. But again, not common in the clinical setting. What's getting more and more common, though, is looking at the DNA. So why would we not always look at DNA? Well, these tests are a little bit more expensive, and sometimes they take more time. If you're not able in your own lab to send things out for sequencing and you have to wait for sequencing results, right? It's sometimes just faster and easier to do a 15-minute COVID test rather than, okay, well, let me try to see like what, RNA I can isolate from the nose and see what virus it is. I can stick a swab up there and know in 15 minutes. And so, what we look at are things like DNA-based composition, for example. We call it the GC ratio. If an organism has a certain percentage of G and C, let's say if you add up G and C and that equals 60%, we know that the combination of A and T is 40% because it has to equal 100. So if G plus C equals 60, A plus T equals 40. If the other organism has G plus C of 40, we know they're probably not closely related because the GC values are very, very different. So you can even look at GC values to see whether or not organisms are closely related. If it's 59 in one cell and 60 in the other, maybe they're really close. But another really cool assay that we can do with DNA is called a restriction fragment length polymorphism assay, RFLP. When we're being really annoying, we call them RIFLIPs because RFLP takes too long to say. This takes advantage of restriction enzymes. What we know is that when you digest the DNA of two different species with the same restriction enzyme, you get a different banding pattern. You get different size fragments because they have different sequences of DNA. If you have two cells of the same species and you digest their DNA with the same restriction enzyme, you should get the same pattern. So you do the digestion of the DNA and you separate on an agarose gel and you compare the banding patterns. This is also a technique that is used relatively commonly in outbreak tracing. If you're trying to determine the source of a bacterial outbreak. The E. coli in this spinach matches the E. coli we found in the spinach packaging plant, which matches the E. coli we found in the field where the spinach was grown. Right? They have the same pattern. They likely, right? The source in the spinach, which we found in people, right? This person has diarrhea. They have E. coli with this pattern. They ate spinach that has this pattern. They got it from this facility, which has this pattern, from this field, which has this pattern, from those pigs, which pooped on the, right? You can follow where the bacteria came from. So if you were to look at this, right? You look at lane 1 and you look at lane 6, they're very, very similar patterns. These are likely the same species. 2 and 3 have similar patterns to each other that are different from 1 and 6. They are likely the same species. 4 and 5, similar to each other, different from the others. They are likely the same species. And 7 is the oddball. 7 is probably the latter, but that's okay. So again, you can do this, and this is actually relatively easy. You digest for a couple hours, run it on a gel for an hour, done. And you didn't have to send it for sequencing. But we can also send for sequencing. So we can do PCR tests. So we can amplify DNA. This is how they were diagnosing COVID, like in clinical labs. They would do a PCR. One of the challenges, though, especially with some of our RNA-based viruses, all of our RNA-based viruses, they're RNA-based. You can't do PCR of RNA. So first they have to convert the RNA to DNA, then they do the PCR of the DNA. But it's more sensitive than the one that scrambles your brain. So if you, I don't know, like I had my kids in 2021, so I had to go and get tested for COVID before they'd let me come to the hospital. I don't know what they were gonna do if I was positive for COVID. I'm like, well, you still have to get these kids out of me, so I don't know what's gonna happen. I had a C-section, so I'm like, the kids have to come out. What are we gonna do? They do a PCR because it's more sensitive, it's more rapid. I don't know, maybe they were gonna put me in like a special room and not come talk to me, I don't know. Which would have been fine. But you can do PCR. The challenge with PCR is it's harder to identify an unknown organism because you have to have those specific primers that will bind to the DNA of interest. So if we're testing specifically for something like COVID, like a particular flu variant, maybe we're testing for Lyme disease, that's easy. If we have no idea what the patient has, just doing a standard PCR can be a little bit not informative. We wouldn't want to do that. But that leads us to, what if we don't know what the patient has, but we know it's a bacterial or a viral or even a eukaryotic parasitic infection? We know it's not a virus, basically. We know it's anything but a virus. Can we do PCR to figure out what it is? And the answer is yes, in combination with sequencing. So what we would do here is what's called ribotyping. We would amplify the ribosomal RNA and send just that little piece of DNA for sequencing. And that can actually be done usually overnight. If you can ship the sequence off early enough in the day, you will get your results back the next day. So we do this type of sequencing where we PCR amplify a gene, and then we want to sequence the one gene. If my student can get it in the mail, usually before 2 o'clock, before like the last UPS pickup, I usually have the sequencing results by 7 the next morning. And the facility we use is in Kentucky, so they just like put it on a plane, send it to Kentucky. Kentucky's like, cool, one gene, psh, and I get my results in less than 24 hours. If they're really slow, it might take 48. So you can get pretty rapid results this way. This could be great for infections that are challenging to culture or take a long time to culture, tuberculosis, or are dangerous to culture. So tuberculosis, Francisella, plague, things that you don't want to culture, things that are hard to culture, treponema, chlamydia, things that you can't culture. So you can still do PCR and a little bit of sequencing in order to identify an organism. And finally, if you work in a real fancy place, again, in either a research lab or a fancy clinical lab, you can do fluorescence microscopy. So again, what you're looking at here are antibodies to a particular pathogen that have a fluorescent tag. You take a patient sample, it could be a biopsy, it could be a swab, whatever, and you incubate it with the fluorescently tagged antibodies, and if you see binding, then you know what species you have. So they call it fluorescence in situ, that's why you can do like a tissue biopsy, in situ hybridization. So there are lots of techniques, and especially those of you going into clinical medicine, that you'll become more and more familiar with. If you take immunology, you'll talk a lot more about serology. Same thing if you take hematology, you'll talk a lot more about how white blood cells work and how pathogens are recognized and identified in the blood. So it's a really important field. But what you have to do then, I've already done it, is you have to make a dichotomous key in order to figure out what unknown organisms you have. So you put all of these classification methods together as much as possible, and you can try to identify your unknown. So the first way is through a dichotomous key. And I know this is one of those things that we always insist that you make. And depending on your lab instructor, sometimes your lab instructor will not even let you start the unknown project until you show your completed dichotomous key. Like they won't give you any tests at all until you show your completed dichotomous key. Sometimes you just have to have started and maybe they'll let you have a couple. I'm actually one of the nicer ones. I'm not teaching the lab, so good luck to you all. But you come up with a dichotomous key. So for example, I actually don't start here. I usually when I make a dichotomous key, because there is a potential that there's at least one acid-fast organism in the unknown mix. That's potential. So I start with acid-fast. Yes or no? Yes, that's pretty easy. There's only one. No, all the rest, right? Then I do gram positive, gram negative. And then for each of the gram positive, gram negative, rod or coccus, right? And then biochemical tests, right? I have my old dichotomous key somewhere that I made one day in lab when I was teaching the lab. But the goal behind a dichotomous key, it's in the name, di. You only have two options. So it can be either gram positive or gram negative. It can't be gram positive, gram negative, or not. Right? That's why I start with acid fast, because that's not gram positive or negative. One of the media that's a little bit tricky is EMB. Because you guys have used EMB, right? There's 3 possible results. It's pink, it's purple, or it's green. So I'm like, okay, trichotomous key. No. So what you do for that one, that's what I do anyway. On EMB, ferments lactose, yes or no? Yes, green or purple. No is pink. So you have 2 levels to get that dichotomous key. But either way, at the end of the day, you're like, okay, I have a gram-negative organism. It is oxidase negative. It does use citrate. It's Klebsiella pneumoniae, right? You can track and figure out what organism you have. You don't need to use all the biochemical tests. There are lots of ways to make a valid key for all of the organisms that you have. But I told you my big trick where I start is with acid fast, then gram stain, then morphology. That's how I make my tree. Okay. So it's that flow chart that allows you to identify the organisms that are in your unknown mixture. So again, you have to ask successive questions that have a yes or no answer, or like a positive, negative. There's only 2 responses. And you start as broadly as possible. Acid fast, gram stain. And then you get more specific. Gram positive, coccus, catalase positive, ferments mannitol, coagulase positive, Staph aureus. You end up getting to that point. Now, what this doesn't give you is phylogeny. This doesn't tell me anything about the relatedness genetically of the organisms. It allows me to determine what my unknown species is. If I want to start to place my organism in the evolutionary context, I can then generate a phylogenetic tree. This is where having some genetic information, especially things like 16S rRNA sequencing, can be really, really helpful, because you just look at how similar species are. So, this species and this species down here are 70% similar. They're the most closely related. They have the highest level of similarity. And then you have 60 and 60, and then 50, 50, 50. So those are more distant. And you use that to build a tree. So in theory, right, the shorter the branches, they really lean into the tree metaphor here. The shorter the branches for each of the leaves, the more closely related the 2 organisms probably are. The longer the path it takes you to get from one species to another, the more distantly related they are. Okay. So you can always create the evolutionary relationships once you start to have, especially the genetic information. And as you're working in the lab, you don't have this one, not the systematic bacteriology, because that's based on rRNA sequencing. You have the Berge's Manual of Determinative Bacteriology. This is the Bible. Is Staph aureus this positive, that positive? It tells you everything. Use it. There's 3 copies. Just grab one for your group and sit there and make your dichotomous key. Really, really important. I think it's Dr. Kong who is very much no, it needs to be finished. But it could be Dr. Tao, I don't remember. So you want to have that dichotomous key finished so you can start your unknown project. All right, questions about classification of microorganisms before we move on to starting our discussion of eukaryotes, with particular emphasis on those that cause disease. Okay, cool. Eukaryotes. Eukaryotes they're okay. They're not my favorite, but that's all right. When we talk about eukaryotes and we talk about microbiology, especially when we talk about microbiology of things that can make you sick, we most often are looking at things like fungi, algae, protozoans, helminths, and sometimes we talk about arthropods. We talk about arthropods for a couple of different reasons. Arthropods are, one, very important disease vectors. They spread a lot of disease. But they can also cause issues with people through things like envenomation. And then you got weirdos like botflies that will literally, you will grow a maggot in your body. So if you've ever met an entomologist, they're sometimes a little bit strange and they like botflies and they're like, oh man, I wish I had a botfly. And I'm like, why? Y'all are something, man. It's like, ooh, I wish I got staph. No, it's gross. But that's cool, whatever. All right. Let's talk about fungi. Fungi are weird and very, very cool and very, very diverse. The field of mycology is the field of scientists who study fungi. They have cell walls made of some sort of carbohydrate, usually chitin, glucan, or mannan. Depends on the fungus. They are what we call chemoheterotrophs. They are not capable of making their own energy. They have to get it from something they consume. And they get their chemicals also from something they consume. So they don't use light energy. They can't fix carbon dioxide. They have to eat something that was already alive. And they are very, very important as saprobes or decomposers of organic material. They are usually either aerobes or facultative anaerobes. They can survive without oxygen, but they prefer oxygen. Depending on the species, some fungi do engage in sexual reproduction, some reproduce asexually, and oftentimes they do reproduction through spores. Spores of fungi are for reproduction. Endospores of bacteria are the dormant stage for survival. So don't confuse them. Even though we typically don't say endospore, we often say bacterial spores. They're different from fungi spores. There is a ton of fungal diversity. It would be really cool if we had a mycologist who could talk all about, fungal diversity. In terms of pathogens, not a ton. Approximately 100 species or so. Every once in a while, we'll discover a new fungus that's capable of causing infection in people, but usually that signals there is something very wrong with the person. Usually it's a complete collapse of the immune system. So when you want to grow fungi in the lab, you might sometimes want to try to culture a fungal pathogen from a patient. Not always. Fungi typically grow a little bit slower than most of the real common bacteria. So one way that you can try to isolate them without having them be outcompeted on the plate by bacteria is by reducing the pH to 5. That often can inhibit some of the common bacteria that might be in the same environment. And also, they're a little bit more resistant to osmotic pressure, so a higher concentration, typically of sugar, but sometimes salt. They can sometimes grow in a little bit lower moisture content, and they can metabolize more complex carbohydrates. That's why they're really good as decomposers, especially of things like wood, why you'll often find mushrooms at the base of like dead or dying trees. Now, in terms of pathogens, often we see pathogenic fungi in the yeast form. So yeast are unicellular. Typically, they're oval in shape, though they can be spherical. This is one of those, like if you have a patient with a bloodstream infection and you see a more oval shape that's actually pretty large, you're often looking at a yeast infection in the blood rather than a bacterial infection in the blood. So yeast, again, typically are facultative anaerobes, right? The classic example is Saccharomyces cerevisiae. When you force it to do fermentation, you get bubbles and booze. Okay? You get the carbon dioxide and the ethanol production, though it would prefer to not have to do that. They can reproduce through fission, where they reproduce evenly, but often they reproduce through budding, which is what's illustrated here. Like one new cell buds off the parent cell, and you can actually, under electron microscopy, see the bud scar that's left when like one bud falls off. So again, typically the yeast stage of many of these fungi is what's going to be causing an infection. But fungi can also be molds or more of a filamentous type of structure. And in that case, we sometimes see that fungi can be dimorphic. They can switch between a yeast and a mold form, and we'll see that on the next slide. But here they're showing you Aspergillus, which is a really common environmental fungus. When you grow it on a plate, it actually forms this mat of fibers called a mycelium. And then it forms, for its spores, it forms these they're called hyphae, that extend upwards, like into the air. And on the very end, you'll have the structure called the conidia that has all the spores in it. And they're black. Aspergillus niger, they're black. They actually can grow pretty tall. The first time I ever taught microbiology for like the nursing students, I had them go around and like look at dust and plate dust to see what they could get. And one of them had just a plate full of this Aspergillus, and she started to open it. And I was like, girl, don't open it. But she started to open it anyway. And the aerial hyphae, the ones extending up, were actually pushing the lid up. So when she like opened the lid, they kind of sprung up, and I was like, ah! Close it. Okay, please don't open the lids in class, right? They can get pretty tall, but this can also get into the body and cause an infection. And they'll form this kind of mat structure. One of the common places they do it is in the lungs. So in your patients that have had things like emphysema, COPD, lung cancer, where they have cavities kind of left over in their lungs, they'll get these, they call it a fungal ball, but they get the fungal ball in there of fungal infection. So again, the pathogenic species often can be dimorphic. They can switch between this mold and this yeast form. And they typically will be the yeast form inside of the body. They replicate more quickly, they can disseminate more easily, but sometimes you can see them do both forms in the body. Like Candida albicans, which is the really, like if you hear about people talk about a yeast infection, they're usually talking about that. It can switch inside the body. It can form the hyphal structures with the elongated, and it can be yeast. But some of our really dangerous fungi, like the one that we have in the valley that causes valley fever, it's usually a mold in the environment, and then it gets inhaled and is a yeast in the body. So fungal infections are called mycoses. They're often a lot more difficult to treat than a bacterial infection. And so in many cases, they become chronic, especially some of the things like, again, valley fever. My very first semester at Fresno State, one of my students was like, my dad has valley fever and he's been on antifungals for 6 months. And I was like, yeah, that sounds about right. And the doctor said he's got at least 3 months to go. Sometimes it's lifetime. If you have patients with HIV that develop fungal infections, they are on antifungals for the rest of their lives. People complain about recurrent yeast infections. They just come back. They're really hard to get rid of. So we'll pick up on Thursday talking more about eukaryotes and the different kinds of diseases that they can cause.